right, everybody, we're going to go through a little primer on hematopoiesis. So this should be a review of the anatomy and physiology of stem cells and how they develop to become into adult blood cells. So we'll go over hematopoiesis in this lecture, which is going to include the formation of erythropoiesis, leukopoiesis, and thrombopoiesis. So hematopoiesis is the term for the creation of blood, the elements of blood, and then those are the component parts that we'll go through. And if you understand what those things are meant to do, things like platelets and red blood cells, then it will make sense when there is a deficiency of them. So then you'll go into the pathologies and we'll learn all of the different disorders that can come about because of abnormalities of hematopoiesis. All right, so just starting off, the blood is made of two main substances we're going to talk about. The first being the plasma, and plasma is mainly water. About 92% of it is water. The remaining is a mix of various products such as gases, any nutrients or drugs or vitamins that are going through the body. There's proteins. The largest or most prevalent is albumin, and then things like electrolytes. And this is something that I want you to think of as the bank for water in the body. So when we need a quick source for water, the plasma is always a nice place to tap into. The remaining elements are the formed elements, so 45% of the blood is suspended in the plasma, and that includes red blood cells or erythrocytes, about 95% of the formed elements are red blood cells, about 5% are platelets, and a small amount, 0.1%, are white blood cells, which then falls into a couple of different categories. We have neutrophils, eosinophils, basophils, monocytes, and lymphocytes, the specific immune cells. So these things all have to form, and they form in their own processes. And you can take a look at what blood looks like when you take a blood sample from a patient. It's spun down in a centrifuge and it separates by weight. So what you'll see is that the plasma is going to end up at the top. Again, it should be about 55% is the perfect amount. Between the next level and what we call the hematocrit here at the bottom, there is going to be a buffy coat, which contains the white blood cells and platelets, and the hard-packed red blood cells that separate as the heaviest at the bottom. We do refer to that as the hematocrit. So that perfect amount of hematocrit should be 45%. And technically, if it's less than 45%, we refer to that as anemia. You will see that it's a little bit more complicated than that, and numbers do shift depending on age and other things about a patient, but that's generally what we're looking at here. And all of these things are derived from the bone marrow, where they form from stem cells into the component parts. So we'll be talking about different types of stem cells that form into these mature formed elements of red blood cells, white blood cells, and platelets. So it begins with a little primer on a stem cell. So there's a couple of different things I'm going to orient you to. So when a sperm and egg gets together, it has the ability to become any single tissue in the body. When we're referring to those stem cells, so it's just a blank slate cell, we call those totipotent stem cells. So they have the ability to become literally any single tissue in the body, including those associated with pregnancy. After there are a couple of different cleavings of these original totipotent stem cells, we get what's referred to as a blastocyst. And a blastocyst is made up of two pieces, for simplicity's sake. We have the outermost layer called the trophoblast here in pink, which is going to turn into eventually a placenta. And then the inner cell mass, for simplicity's sake, will become everything associated with first an embryo and then a fetus. So these cells of the inner cell mass have the ability to differentiate into any tissue in the body, but not any tissue in pregnancy. They're going to be the things that make up the tissues in the body. So really the only difference here, a totipotent stem cell can become any cell in the body, including pregnancy-derived cells, whereas the next level, we call it a pluripotent stem cell. So it has the ability to turn into any adult body cell. So anything that you have in the body came from these pluripotent stem cells. And once we are at the level of the body, so somatic means body, soma is body, literally, pluripotent somatic stem cells are body-related, what some people call adult stem cells. So they're in the body and they are waiting for different growth signals so they can go forward and change and mature into adult tissue cells of the central nervous system, peripheral, hematopoietic system, which we're talking about today, liver, skin, mesentery, etc. So all of these cells are derived from an original stem cell. And we have them all over in the body, and that's what we're going to talk about with hematopoiesis. So these stem cells, which are in the bone marrow, they are waiting for a chemical signal to tell them to go forward and differentiate. So we're going to start with a stem cell, and when a stem cell starts dividing, after it gets a message that it's going to become something else, the daughter cell, or family cell, is called a progenitor cell, and that comes from the word progeny, which refers to like your children, for example, your progeny, so they are derived from you. So stem cells become progenitor cells, and those progenitor cells go through their own process to become a mature formed element. So depending upon which chemical signal is received by these blank slate stem cells in the bone marrow, they can be derived into any of these adult tissue cells. And that's what you look at here at this slide. Hematopoiesis is the formation of these adult tissue types. So we've got our specialized immune cells, T cells, B cells, plasma cells, natural killer cells. Those are the immune types of cells. And then all of the other ones are derived from the myeloid progenitor family. So starting off just very simply, the original multipotential, what we call an adult stem cell. This is a multipotential hematopoietic stem cell, or a hemocytoblast. Hemo is blood, cyto is cell, and blast refers to an immature cell. It's not fully evolved yet. It hasn't become completely matured. So these adult hematopoietic stem cells, they can turn into, by differentiating, into any of these mature formed elements. The other thing special about these hemocytoblast cells is if they don't receive a chemical signal to go forward and become something, they can also just split and become more stem cells. So they have the power of self-renewal, where none of the other cells on the slide do. Again, depending on which message is received by these cells, they will become a progenitor cell and go down the line of progeny. Once a cell is a progenitor cell, it now does not have the ability to make a copy of itself, so it's lacking the power of self-renewal. It would also say it's committed to that cell line. So once we have a myeloid progenitor cell deriving from a hemocytoblast, we can't have this myeloid cell suddenly change to become a natural killer cell. It now has to belong to this family, so it is committed. So it can't become more of itself, and it has to become something in that family. So all of these mature formed elements, they can be traced back to this single progenitor cell, which derived from this adult stem cell, which is in the bone marrow. So the formation of red blood cells is called erythropoiesis. And in this process, we're just looking at a form of linked mitosis, where the stem cells, they go through these various iterations, so they're still splitting, but every time they do that, they look a little bit less and less like the original parent cell. So the stem cell becomes a committed progeny cell, and then going through this process, you'll see that eventually it spits out its nucleus, and the step before we have a mature red blood cell is called a reticulocyte. So sometimes you'll see tests for reticulocyte counts. And eventually, the mature red blood cell is going to be produced. So again, this is starting with the stem cell, and then it goes through these various processes to become this mature formed element. It starts to get some hemoglobin, and then it spits out its nucleus, and now it's mature. And red blood cells, as we saw, they're the most prevalent mature formed element. They look like little Paula's donuts waiting to be filled with delicious cream filling. And that small shape makes it easy for them to move through the microcirculation and squeeze through places without obstructing anything. So having them in this natural shape is really important. So they're biconcave, meaning there's two invaginations, one on each side. They don't have a nucleus because when they are mature, they have already extruded it in this mitotic process. They have hemoglobin associated with them. A normal, healthy red blood cell with a healthy spleen in that person too should live about 120 days. The bone marrow has to continually be making stem cells essentially divide into mature red blood cells to make up for the percent that's cleared by the spleen at the rate of about 1% a day. So there's a huge amount of mitotic capability of cells destined to become erythrocytes. And in order to make red blood cells, this is important for the pathology, there is an absolute need for B12 and folic acid. A lack of either one of those will lead to abnormal blood cell development. Here you're looking at a heme group, and at the center we have a little iron. The hemoglobin is a globular protein structure, so that's made up of these 4 globular subunits, and then the heme group at the center with an iron, and each little iron can bind about 4 little individual oxygen molecules. So that's what this iron looks like. So you probably know that iron deficiencies are associated with things like anemia. That's because we need this iron to be able to bind oxygen. Another really important thing about this iron ring is that if it had its choice, it wouldn't bind oxygen, it would bind carbon monoxide. The attraction or affinity for carbon dioxide, or monoxide, to this iron ring is 250 times greater than it is for oxygen. So keep that in mind with regards to carbon monoxide poisoning. When there is the creation of red blood cells, it involves a signal called erythropoietin, or EPO. And EPO is created whenever there is a reduction in the amount of circulating red blood cells. So that could be due to decreased blood flow, so maybe there's some cardiac abnormalities or vessel abnormalities. It could be due to COPD, respiratory disease, so there's obstruction. It could be due to being at a high altitude, decreased levels of oxygen in the blood. They're going to signal the need for more EPO. If somebody's bleeding, so remember GI bleeding, really common with colon cancers, when there's chronic blood loss, like GI bleeding, or there's decreased hemoglobin formation, all of these things are either going to decrease oxygen delivery in some way. Then that's going to signal to the kidneys to spit out this product of EPO. Whenever there's EPO, there's more erythropoiesis, there's more red blood cells, and there's more of an ability to bind oxygen. This is a slide that's showing you all of the different processes in each of these tissue types, and I want you to notice that the red blood cells are the only thing that require EPO. The other families just require generic growth factors. Erythrocytes, EPO, erythropoietin is the signal to go from a progenitor to a reticulocyte, eventually to the mature red blood cell. You've probably heard of this with blood doping. That's what that refers to, is the intentional taking of EPO. And you can think about how awesome that would be for somebody like Lance Armstrong, who was convicted of blood doping. When you give yourself lots of EPO, you make a lot more red blood cells, which means you really don't have to ever make a shift from aerobic to anaerobic metabolism. So therefore, you never fatigue. How great is it to never fatigue? You'd be really good and win a lot of Tour de France's. So again, this should only be signaled when there's an imbalance at a homeostatic level of the ability of red blood cells to deliver oxygen to tissues. So the kidneys do release EPO to stimulate the bone marrow to start making more red blood cells to achieve homeostasis. So we will see alterations in homeostasis in the pathology. Where do red blood cells go? They go eventually to the spleen. The spleen loves red blood cells. It eats them up and it breaks them into little pieces. And an important thing to remember about the spleen is the bigger and more delicious or weird looking the red blood cell is, the spleen will eat it even more quickly. So normal red blood cells, again, they're cleared about 1% a day. Abnormal blood cells, the spleen will gobble them up immediately. And the spleen breaks down red blood cells into a couple components. The first is iron. That iron ring is popped off, and then a transfer protein called transferrin stores that iron as ferritin. That means that we don't have to have a continual replacement of iron from broken down red blood cells because it's stored in the body. The globular portion is going to be broken down into amino acids, and then the heme portion will be broken down into bilirubin, which you probably have heard of before. It's what makes your skin look yellow and causes a presentation referred to as jaundice. And in order for bilirubin to leave the body, the liver has to conjugate it, which means it has to add something to it to make it water soluble. So the liver has to be active to do the conjugation response. And the other thing the liver needs to be able to do is make bile, because bile is released when we're incorporating something into the feces. And the feces are the way that bile leaves the body. So the liver has to be active and it has to have a free open duct so that it can deliver bile. Okay, so this is the just process for iron recycling. Again, there's not a lot that is required for replacement because the body will recycle as much iron as possible. That should tip you off for things like iron deficiency anemia. In adults, it usually means that somebody's bleeding somewhere. Okay, and then just going through very quickly, white blood cells. Again, we've got our granular cells, agranular cells. Each of these has its own little function, and generally they just are formed from a common precursor. Whenever you see increased white blood cells, it means that somebody's fighting an infection. There's something that is making them ill. Here's our agranular, which means no granules when you stain them, and granular do have visible granules, which can contain a variety of substances like histamine. So neutrophils are cells that are thought to be designed to fight bacteria and fungi. Eosinophils are cells that are really good at fighting larger parasites, but they're also responsible for things like allergic inflammatory responses that make you feel really miserable when you bind an allergen. Basophils include things like mast cells. They release histamine. Lymphocytes are the specialized cells of the immune system, B, T, natural killer cells, and monocytes are the tissue-specific macrophages. So most of the time there are specialized just monocytes in each area. It's kind of like a security guard in each area. They just take care of local infections in that tissue, and then the whole team is called in when things get really bad. The formation of white blood cells is called leukopoiesis. It begins with a hemocytoblast. This includes cells that are going to eventually become immune cells. So we have a lymphoid progenitor here, whereas we also have things like neutrophils, basophils, and eosinophils. So those will utilize a myeloid progenitor. As I pointed out before, a growth factor is what is needed to go forward in these pathways. There's no EPO, and they just have their own development process. So they start as a stem cell and eventually become a mature white blood cell. Finally, we have platelet creation, and platelets are called thrombocytes to help you remember that they're really, really good at forming a clot. Thrombo means clot, and poiesis is formation. So the formation of platelets is a little bit different than the other types of linked mitosis you saw with red blood cells and white blood cells. When a platelet is being created, we start again with the stem cell. there's a myeloid progenitor, and then it becomes a really large precursor cell called a megakaryocyte. And megakaryocytes, they grow and they grow and they grow, and the more they grow, the more they flake off little pieces of themselves. And those are the platelets. Isn't that fun? So they come from these megakaryocytes in the bone marrow that just start to get really big and then shed off little pieces of themselves. So that's called fragmentation, little fragments of them uh, essentially shed off. So it looks granulated as it's developed, and it's the only one that is going to use this fragmentation process. So those are the different types of hematopoiesis, and feel free to watch the next lecture on the pathology.